All right, gene expression, quick and dirty. So we've been talking about specialized cells. That was kind of the framework. How do our cells become so different from each other when they all have the same genetic material? And then I gave you some background on biomolecules and cells themselves. Now let's go back to gene expression. So how does that DNA become these different proteins that then make our cells different? So let's go to the genetic material. So here is another image of the cell, a cell with a nucleus. Inside the nucleus, there's a bunch of DNA. DNA is packaged up into chromosomes. Humans have 23 pairs of chromosomes that often are depicted like this, although they're often not in that structure unless the cell is dividing. Chromosomes, the important part for us is that it contains double-stranded DNA. The DNA is actually wrapped around proteins to be packaged. Just another example of proteins being awesome, the histone proteins. Other than that, we're not going to talk about DNA packaging much, even though it's awesome. So this DNA double helix is made up of nucleotides and contains the code, the information to make up any gene that you have. Humans have about 20,000 genes that code for proteins. There's other parts of DNA that are also important. That is the topic of another course. So we talked about nucleotides themselves, that they make up, nucleic acids such as DNA. And the basic central dogma is that this DNA is copied into RNA and then translated into a protein language. So to review that, here is our genetic code and then the overview of central dogma. DNA, RNA to protein is central dogma. So we've got this code that's the DNA, the language of the DNA, and that is read into RNA. That process, I'm not going to review. RNA is read in, not the entire DNA is made into RNA in all cells. That RNA is then translated into protein. So translation is because it's a new language. Proteins are made from the linking of multiple amino acids. Again, those building blocks, the monomers that make up the polymer, which is the polypeptide that then folds into a protein. And so this is going to happen, this process, differently in different cells. Your neurons are going to make different proteins, different sections of DNA will be made than your red blood cells. Your red blood cells are going to make a lot of hemoglobin protein, and your neurons are going to make neural proteins. So this protein is made, and then let's talk about protein structure. The protein is made just as a long string of amino acids. This is the primary structure of the protein, and this is important because it's going to determine how the protein folds. Secondary structure are these alpha helices and beta sheets that fold based on hydrogen bonding. Tertiary structure is really where we're going to be looking. That's the folded protein, three-dimensional, either globular or fibrous, various shapes, all kinds of shapes that have different functions. But that tertiary structure is where we can start to have function. So something about this protein right here, this is showing transthyretin, which is able to bind thyroid hormone. Something about its structure is able to bind this thyroid hormone and also travel throughout the blood easily. Quaternary structure is present for some proteins in order for them to function. Not all proteins have quaternary structure. Hemoglobin is an example of quaternary structure. There are 4 subunits to hemoglobin. So the primary structure of the protein is going to determine the tertiary quaternary structure, therefore the function. And of course, the DNA structure code is going to determine the protein structure. So here's an example of how a change in the DNA can alter protein function. Not every mutation has a visible or phenotypic, effect. There's some redundancy in our code in terms of mutations that are silent, they don't matter. There's also certain amino acids that act very similar. This is a nice example because it's a mutation that then results in a very different amino acid with different properties that causes the protein to fold differently. So sickle cell anemia is something you may have heard of. It actually is such that a single copy of the sickle cell gene is actually somewhat advantageous to be heterozygous. There's less susceptibility to malaria. If you have two copies of this mutant, though, you then have a problem because this protein does not fold how it normally does. So this is a single base mutation in the DNA that is then a different mutation base in the RNA. And in this case, it results in a different amino acid, and the protein is different. So the protein in the sickle cells is clumped. It clumps together because it has a different shape to it. This is our quaternary structure of hemoglobin, composed of 4 subunits. This is going to result in the cells actually being sickle-shaped themselves as well. So it's a really nice example of structure-function relationships because the structure of the DNA determines the structure of not only the protein but the cells, and it's kind of a cascade. Now, over evolutionary time, this results in diversity, right? So mutations can cause diversity advantageous traits to evolve. So just kind of keep that in mind. So let's go over to some types of proteins. So kind of getting back to the idea of how can we have these different proteins produced in cells that are genetically identical, and why do those different proteins matter? Like, how does that matter? So, for example, membrane proteins are going to be really important on all cells, really. But let's just say neurons as an example. Neurons need to be able to communicate. So if we had a membrane protein that was a receptor, it would bind the neurotransmitter that's released from the cell that's talking to it. And it might have certain membrane proteins. You've heard of glutamate, maybe, or dopamine. It might have a receptor. If there's one dopamine cell talking to another cell, that cell it's talking to needs to have a receptor, a special membrane protein that can detect that dopamine that's being talked to. There's keratin and collagen are 2 types of fibrous proteins. So they're fibers, they're long instead of globular, that are present in things like hair and skin. And those proteins are present there because the genes are expressed more so keratin is in your skin. It's not produced in your red blood cells. That protein is not made. If it was, your red blood cells would not function correctly. Hemoglobin, on the other hand, is what's present in red blood cells in large amounts. So that protein's produced. Antibodies fight infection, enzymes, tons of different kinds of both enzymes and antibodies, tons of kinds of membrane proteins. These are examples of the actual structures besides the membrane protein, what the proteins actually look like in terms of their structures that allow them then to function. So this enzyme would be specific to, I'm actually not sure what enzyme this is, but it's going to be specific to one type of, one substrate based on its structure. So the 2 main types here are fibrous and globular, but the main point is it's a huge diversity of proteins out there. We will see many examples this semester, and they basically do everything, right? So enzymes catalyze reactions, transport is membrane proteins, as well as hemoglobin, transport of oxygen, and communication, so various types of communication. There is one, I don't have that. Okay, oh, here it is. This is a really nice website that I recommend. I have it posted as well on your Canvas page that kind of breaks it down by types of proteins a different way. It's going to load here in a moment. So different enzymes, structural proteins, signaling, sensory, transport, regulatory, motor, defense, and storage. Some of these a little bit overlap, that's okay. So let's go to structural proteins. It's going to bring up several examples of structural proteins. Collagen is one that is a part of a lot of connective tissue we'll see. So this is a nice website for kind of a way of categorizing and seeing examples of proteins. Here, signaling, we've got an adrenergic is binds to adrenaline with epinephrine, and insulin binds to insulin receptor. So some examples of signaling molecules. All right, so the big idea here again was that these proteins are present in different amounts in different cell types. And this is what allows our different cells and therefore our different tissues to function differently and be specialized and make you special.